Hi everybody, Dr. Ahles here. In this short video, we're going to talk about a structure that we find in our chromosomes called telomeres. When we talk about telomeres, we need to consider the fact that every time your DNA is copied, it actually gets a little bit shorter. This is because when we first copy our DNA, remember that we added an RNA primer to it, and that RNA primer is where I started to copy my genetic information. But adding that RNA primer leaves a little space where I didn't copy the nucleotides ahead of it. And so when I copy my genetic information, I miss a few nucleotides each time I copy it, meaning my copy got shorter and my copy of my copy got shorter, and slowly over time, because I have to keep adding primers, my DNA sequences get smaller and smaller. When we think about copying our DNA, it's a little bit scary to consider that some of our information may not make it from one copy to the next. This is why our chromosomes have structures at the end of them called telomeres. Telomeres are places of repeating nucleotides. These nucleotides don't code for an important protein in your body. They don't do anything except kind of act as a buffer at the end of your chromosomes. So when we replicate our DNA, these telomeres, these repeating regions at the end of our chromosomes, will slowly get shorter and shorter over time. But because those telomeres aren't vital information, because they're just repeating regions of DNA, all of our important genes are still intact. That being said, with those telomeres getting shorter and shorter with each division, this is what may be leading to a lot of the aging effects that we see in our cells. So cells that become less functional, cells that get smaller in size, a lot of those kinds of changes may be due to the loss of telomeres. Now, because telomeres are so important for making sure that all of our DNA gets copied, we have an enzyme called telomerase, whose job is to make sure that the telomeres stay long, or to extend the length of our telomeres, especially in cells that are dividing a lot. So for example, sperm and egg cells, we make sure that telomerase is very active, making our telomeres longer. So notice I have a DNA sequence up here. This DNA sequence, when it's copied, would normally start at this position, and we'd copy to the left. We'd lose this information. What telomerase does is it comes in and adds more copies of the same repeat to that region. By adding more copies of the repeat to that region, some of that information that I would have previously lost is now being copied. So telomerase makes these repeating regions longer. By making them longer, our important information gets passed along. So we add more repeats, everything after that is saved. So consider a cell like a germline cell, like I mentioned, sperm or egg. These have telomerase, adding these extra bases at the end, whereas a somatic cell or a normal cell may not have all of that telomere left. In that case, this cell is going to start with every single copy having its DNA be shorter and shorter. This is going to lead to aging, whereas having that extra telomere keeps those cells healthy and passing along all of their genetic information.